Let's now focus on viral structure, genome structure, and viral replication. We already discussed the structure. HIV is an envelope virus where the viral envelope is derived from cell membrane. Within the envelope, there are glycoproteins, those surface units, which are binding glycoproteins. Those are glycoproteins which bind to the target cell receptor, and they are forming trimers. So they are GP120 surface glycoproteins. Transmembrane fusion protein GP41 mediate fusion of viral membrane with target cell membrane. You can see matrix, capsid, which has this very typical bullet shape, and two RNA molecule, reverse transcriptase protease, VPR, small nucleocapsid, etc. Now, the viral genome, this is provirus. As you can see, there are two LTR. You recall LTRs are generated during retrotranscription when virus synthesize double-stranded DNA. GAG, POL, ENV, and accessory proteins. Recall GAG, group-specific antigen, encode for core protein, matrix protein, and small nuclear protein. POL region polymerase encodes protease, reverse transcriptase, and integrase enzyme, and ENV encodes GP120 and GP41. These three groups of genes are common for all retroviruses. However, as we said before, HIV also encodes accessory proteins, which play a very important role. Let's start with TAT, transactivator. You can see TAT on 5 and 3 prime end of envelope, so it means it will be spliced. And it is very important to regulate transcription to assure that viral RNA is transcribed efficiently. RAB, which is also spliced upon transcription, plays a very important role in the export of unspliced transcript. You know that in eukaryotic cells, splicing is critical for mRNA to be transported into cytoplasm. However, unspliced transcript would not be normally transported by cellular machinery, so virus encodes RAB, which enables their transport from nucleus to cytoplasm. WIF is viral infectivity factor right here. It plays a very important role in inactivating host cell defense. You recall that cells such as myeloid cell, like macrophages, they do encode and they carry in cytoplasm apobex protein. In this case, particularly apobex3G, which is cited in deaminase. The cytidine deaminase would introduce mutations into viral DNA during reverse transcription process and therefore makes the viral DNA non-functional. So what VIF does, it does targets apobex3G4 degradation. VPR is one of those proteins which carry NLS, nuclear localization signal, and therefore helps the virus to get into nucleus in non-dividing cells, such as terminally differentiated macrophages. It also triggers cell arrest, cell cycle arrest in G2, which triggers much higher viral production. VPU, right here, VPU promotes intracellular degradation of CD4, so it decreases expression of CD4 by enabling it to get expressed. Another very important protein is NAF. It was called negative regulation factor, however, it is not affecting virus negatively, opposite. It augments viral replication in vivo and in vitro. And in addition, it downregulates from the surface CD4 and also MHC class 1 and 2. And you recall that MHC class 1 is critical for recognition of infected cells by CD8. So this downregulation enables the infected cells to escape immune surveillance. Now, let's talk about some major characteristics. We already discussed major targets of HIV, CD4, T lymphocyte, and macrophages. Both of these targets carry CD4 cells and also co-receptors, which are required for viral entry, which are CCR5 and CXCR4. They are both chemokine receptors, and chemokines, natural ligands for this receptor, can block viral entry into the target cells. Now, once the virus enters cells and it integrates into the cells, it can stay latent. However, how it can get into the non-dividing cells, such as macrophages, we know that tissue macrophages are terminally differentiated, so they do not proliferate. It is because several protein, I repeat again, matrix, VPR, and integrase carry NLS nuclear localization signal, which allows them to dock on important transport machinery and get transported into nucleus as pre-integration complexes. Now, once virus integrate, it can stay latent or it can actively transcribe and produce new variants. All this depends on the activation of the transcription factor NF-kappa B and NF-AT. If cell is silent, if cell does not replicate, for example, HIV infects CD4's effector cells, which are actively replicating, so virus can get into nucleus, integrates, but afterwards part of the cell become memory cells, so they stop replicate. So they enter to G0 phase. So in this case, provirus is still there, but it does not replicate. It is latent. And actually, latency is one of the problems why we cannot eliminate the virus from infected cells. Now, when this memory cells is reactivated, so it engaged T cell receptor and activation signals gets into cells, cells start to proliferate, virus can start to transcribe and replicate. Let's now focus on viral entry and replication. So this is a model of entry. We know that firstly, HIV GP120 binds to CD4. CD4 is required, but not sufficient receptors. This first binding is required to trigger conformational changes in GP120, which opens binding site for chemokine co-receptor CCR5 or CXCR4. Once virus binds to chemokine receptor, it further triggers further conformation and enable GP41 to change conformation and insert its hydrophobic portion into target cell membrane. This triggers fusion of viral membrane with target cell membrane, and nucleocapsid, viral nucleocapsid, can enter the cells. Now, there is something very interesting. We already mentioned that co-receptor for HIV are CCR5 or CXCR4. They are both expressed on both T cells and macrophages. However, during transmission from person to person, only CCR5 viruses can establish infection in the new target. So if you isolate the virus from person who was just infected, and during the clinically latent phase, there would be predominant CCR5 viruses. After transmission, you will only isolate CCR5 using viruses. However, later on, during the clinically latent stage, as the pathogenesis progress, mutations are introduced and CXCR4 using strains start to occur. First, there are so-called dual tropic, which can use both CCR5 and CXCR4, but later on, CXCR4 strain occur, and during final stage, its stage, you will see both CCR5 and CXCR4 using viruses. Why this is important? CCR5 are important for transmission. However, CXCR4, when they occur, it's a bad sign because they are much more pathogenic. They replicate to higher level and cause cytopathic effects in the infected T lymphocytes. So they kill much more rapidly and to higher extent the infected cells and CCR5. So occurrence of CXCR4 using strain is always a bad signs. It's bad prognosis. So I mentioned that CCR5 using viruses are one which occur upon transmission or are one which establish infection in new host. And you might have heard that there are some people who are resistant to HIV infection via natural route, meaning via sexual contact. And it is because they carry mutation in the gene for CCR5, so-called CCR5 delta 32, meaning that the gene from CCR5 in these individuals has deletion of 32 base pair within the CCR5 region. And a normal CCR5 receptor, right here, chemokine receptor, is 7 transmembrane domain G protein coupled receptors. If there is deletion of the 32 base pair, basically it triggers synthesis of this kind of receptor, which is non-functional. It cannot be even expressed on the membrane. It's not functional. So if person is homozygous for this receptor, those people are resistant to sexually transmitted HIV infection. We can detect actually this mutation by designing primers which span this region where the deletion occur. And you see here on the left on the gel, this is wild type, which has both functional CCR5 genes. This is in the middle is heterozygous, one functional allele, one mutated, and on right we have homozygous. Those who are heterozygous can get infected by HIV. However, the course of infection is slower and pathogenesis occurs much later than in people who carry wild type alleles. I stress that this deletion only protect against HIV upon sexual contact. It is. There were several cases where people who were homozygous for Delta 32 got infected with CXCR4 using viruses. Those are not transmitted via sexual route. However, those people were intravenous drug users. So basically, the infected blood was introduced directly into circulation, so it didn't go through mucosal surfaces. Even in this case, there are only few cases like that. So even in an intracellular drug user, expression of CCR5 is important. However, in that case, those cases, it does not protect 100%. Now, let's look for replication. We already talked about the binding and fusion. In many older papers and textbook, you will see that upon fusion, nucleocapsid disintegrates. There is uncoating directly upon entry. This is not correct. It was shown several years ago that virus does not uncoat immediately upon entry to the target cells. However, the capsid docks on the tubulin via, you remember, dynein, molecular motor, which walks along the microtubules. And by hijacking this transport machinery, viral nucleocapsid is traveling towards the nucleus. During this time, the viral RNA is retrotranscribed into double-stranded viral DNA. This is completed, this retrotranscription is completed around the time when virus reaches close proximity of nucleus, and completion of reverse transcription triggers uncoating. So virus uncoats in the close proximity of the nucleus. And pre-integration complexes, which contains viral DNA, integrase, matrix protein, VPR, and some other accessory protein are targeted into the nucleus where virus integrase mediate integration into the genome. And as you recall, this provirus, integrated provirus, can stay latent, or if virus is active and proliferate, it is transcribed, and it produces mRNA, which are transported from nucleus to the cytoplasm. Viral proteins are synthesized. A viral GP120, respectively, it is synthesized as polyprotein GP160, which contains both GP120 and 41. So GP160 is targeted to membrane, and where it assembled, GAC-POL and a viral protein are assembled, and a viral RNA as well. And virus is budding out of the cells, and first it is released as immature virion. Why it is immature? Because it still contains polyproteins, which are not functional. Only when a virus is released from target cells, the viral proteases are activated, and only afterwards they cleave those polyproteins into the final functional proteins. So you will be able to distinguish immature virion with mature, because immature virion do not form this very typical capsid. And the last thing is evolution. So where does HIV come from? We know that HIV, by evolutionary clocks, by sequencing the viral strain circulating now, and those which are stored in freezers from 50s, 60s, and those obtained from chimpanzees and old world monkeys, we know that HIV jumped into human population somewhere at the beginning of 20th century. The span is between 1930 up to 1950. And it jumped into human population from SIV virus, chimpanzees or gorilla. And basically, SIV and chimpanzee, SIV CPZ, also trigger AIDS-like disease in chimpanzees. And by analyzing their SIV virus, it was shown that it's also recombinant virus, which jumped into chimpanzee not so long ago, around 100 years ago. So they still didn't adapt it to it. When virus jumped to human population, it, of course, mutated and adapted to humans, and turn from SIV to HIV. How this could happen? We know that in rural areas in Africa, the local tribe hunts so-called bushmeat. They eat chimpanzees and other wild animals, and once you do hunting, when they hunt, there is blood, and if they have cuts or tears, so the virus can actually jump from the prey to the hunter. That's how virus got into human population. Let's go back. So how actually SIV was introduced into chimpanzees? Basically, there are so-called old world monkeys, red-capped mangabeys, greater spoon-nosed monkeys, and which actually infected chimpanzees. Chimpanzees hunt for small monkeys, so the similar mode of transmission could occur as these viruses recombined and created SIV virus and adapted to SIV, And from SIV, from chimpanzee, it actually got transmitted to human. It didn't happen once. There were many different transmission. However, the virus did not have chance. It had to adapt and trigger some mutation to allow it to replicate in humans.